Okay, so everyone, we are going to be covering chapter 6, amino acids and proteins. And in this lecture, we're going to explore how amino acids serve as the basic building blocks of proteins and how their chemical structures determine a protein's shape and function. We're also going to connect these ideas to a real world life application in medicine. And this should show us how even a single amino acid change can lead to diseases such as sickle cell anemia or cystic fibrosis. So just an overview, proteins are incredibly versatile molecules in the body. They transport molecules, they allow cells to adhere to one another, they act as hormones and enzymes, they are structural elements, and they also compose the membrane. Every one of these roles depends on the unique arrangement of amino acids within each protein. The sequence and chemistry of these amino acids determine whether the protein goes in the body and what it can do. A great example of that would be when a protein misfolds or is missing a key amino acid, it can lose its function, like the CFTR protein in cystic fibrosis. So proteins are extremely important when we're talking about medicine and just overall disease. So in slides 3 through 5, there are case studies of patients that some are new and some you have seen before. So we have Will Sickle, who is a 17-year-old that has sickle cell anemia. And the case study, I want you to pause and read it and then come back as I'll continue to lecture. But you'll see as you read the case study on this patient, it gives background on his hemoglobin levels and some other tests that were taken and x-rays that were done to help explain what his ailment is and some of the symptoms he's dealing with. The next two patients are Cal Cullis and diabetes. We are very familiar with diabetes now. She is not a new patient for us. We've seen her since the first chapter here. But Cal Cullis is a new patient. He's an 18-year-old boy that was brought to the hospital with severe pain. And there were tests that were run and x-rays that were also taken. So I want you to take the time to read through these. Think about, as you're reading through these various case studies of these patients, what are some things that stand out to you given what we've already lectured about from chapters 4 and 5, and what are some questions that you have of unknowns. And then the last patient is angina. She's also a new patient for you. She's 54 years old, and they give also some more background about Miss Gina, but it's giving you some information about what symptoms she experienced when she came to the hospital emergency room and what tests they did to help understand what she was dealing with. Now, a clear connection between all of those patients and what we're going to talk about today is the amino acids. Now, you've learned about amino acids in your undergraduate studies, whether it was in general biochemistry, genetics, and even biochemistry. But we're going to give a basic background, and I hope that you gain a greater appreciation for these amino acids. Now, all 20 amino acids share the same backbone. That is an amino group, a carboxyl group, a central alpha carbon, and an R group or side chain. It is the R group that gives them, each amino acid, its own personality, its own unique identity, and that determines whether it's charged, polar, hydrophobic, hydrophilic. So that R group really gives each amino acid its own unique characteristic that really helps determine how it is used within a protein. An example of that would be drug molecules that are produced. They often mimic or bind to specific amino acid side chains. So understanding these groups about these R groups with enzymes and amino acids are important for enzyme interaction. At physiological pH, amino acids exist as zitterons. They carry a positive and a negative charge, allowing them to dissolve easily in water. This should not be your first time hearing about this, because we've talked about this before. While we didn't talk about it from an amino acid perspective, we have talked about an instance where a molecule has a polar or positive end and a negative end, which show dipolar behavior. The exception to these amino acids is glycine, right? All of them are chiral, meaning that they can exist in mirror images of each other. In humans, only the L form is present. But one thing I want to point out to you is when you're looking at these amino acids that show Zwitter ions, all of them are able to dissolve in water because they have that dipolar characteristic. Now you're seeing here on this slide these different amino acids and how they can exist in different forms. All of them are going to exist in the L form in humans. The D form exists in a different state, sometimes in like bacterial cell walls and things like that, but the most common form in mammals is the L form. Now, why is this important? This matters because enzymes recognize the correct orientation. And if you remember from general chemistry, even though you can have a molecule, two molecules with the same chemical structure, how they're oriented makes a difference in how they chemically interact with other molecules. So understanding that is crucial to being able to understand how amino acids function and how they interact with other molecules. So what is the structure of an amino acid? You have the amine group here, you have the carboxyl group here, and then you have the R group. The R group is attached to that central carbon, which has a hydrogen molecule attached to it. Now, a key component of forming an amino acid or protein is joining two amino acids together. And this formation of joining two amino acids together via a peptide bond occurs between the oxygen in the carboxyl group of one amino acid and the hydrogen in the amino group of another amino acid. And together they release water, and hopefully you all remember the formation of these two molecules coming together forms water, and that is a dehydration reaction. Okay, once they come together and they form, that interaction here is a peptide bond. And this continual formation of a peptide bond is how we get the linear chain of amino acids, or also known as the primary structure of a protein. Now, when I talk about the primary structure of a protein, that does not take into consideration any folding or any other types of interaction. That is just the formation of a linear chain of amino acids via the formation of a peptide bond. But remember, I talked about those side chains, the R group. Those R groups can interact with one another via whether it's a hydrophobic region, electrostatic, van der Waals forces, disulfide bonds. Those R groups can interact with one another and form a multi-structured or a 3D structure protein. This is where we get our tertiary structure of proteins from. So you can see here the different color balls you see here are schematic to kind of help you visually see how proteins are formed. The R groups of one amino acid can interact with an R group of another amino acid, and it could be 30 or 40 amino acids away, but that interaction will cause the protein to fold and bend in such a way that you get this three-dimensional structure. And that is how you get the formation of domains with active sites within a protein. Now, remember we talked about the pKa of an acid, right? There are two characteristics that are helpful in determining the classification of amino acids, right? One is the pKa. It reflects how the strength of an acid, but also the hydrophobic index. And that is a scale that determines hydrophobicity. The more positive, the greater the tendency to cluster with a non-polar molecule, okay? All right. And this diagram that you see here is table 6.1. So for those that don't have the textbook, this should help you. You need to learn these amino acids. If you haven't learned them before, or maybe you learned them in undergrad, but then you decided you were going to data dump it and not remember them, now is your opportunity to relearn them. You need to learn these amino acids because they're going to come back again, whether it's in MCAT or whether it is in graduate school or even in your first year of med school. You're going to have to know these amino acids. So take the time to learn these 20 amino acids. Now, they are classified based off of their structure and their interactive or reactivity, but they also have abbreviations. Their abbreviations could be 3-letter or 1-letter. For example, alanine has a 3-letter abbreviation as ALA, and it's also denoted by 1 letter, and that is alanine, or A. So if you were to look on BLAST on NCI and look up a protein sequence, you're not going to see their names written out. You will see a one-letter abbreviation. So it's important that you know what these one-letter abbreviations are. Some are more obvious than others. For an example, glutamine. The three-letter abbreviation for glutamine is GLN, but its one letter is Q. It's not G. G is denoted for glycine. So I just want to make sure I point that out to you so you're aware of the expectations of learning the amino acids, but also the corresponding abbreviations. Another example again is tryptophan. Tryptophan has a three-letter abbreviation of TRP, but its one-letter abbreviation is a W. So do not always assume that the one-letter abbreviation is associated with the first letter of its name, because that is not the case. All right. So how is this relevant to what we're talking about in biochem and the clinical aspect of it? Even if you make a mutation or a mutation is induced and it has just one change in amino acid, that altered sequence can cause diseases like sickle cell anemia, where that one mutation that changed it from glutamic acid to valine formed or caused the hemoglobin shape to change, and it also impacts how efficient it's able to carry oxygen throughout the body. So one small mutation, one base change, can alter an amino acid, and because the amino acids are chemically different or respond differently, it causes a change in the protein shape. So understanding this is also important. So here you have your interaction, so you can visually see the structure. Now, while I will not ask you to draw out the chemical structure of these amino acids, you do need to be aware of these amino acids and how they're classified. The best way to start with learning amino acids is to start with the simplest amino acid, and that is glycine, as you see here. It has the same backbone, the carboxyl group, the amine group, and the essential or alpha carbon with the hydrogen, but off of that alpha carbon is another hydrogen molecule. That is the simplest amino acid. Everyone should know that one. But the next level from glycine is alanine. It's the same structure. The only difference is instead of just having the oxygen, there's another carbon or a methyl group that has been added off of that alpha carbon. Okay, and you'll look here. You have valine, leucine, isoleucine. They all have a carbon backbone that is branched off of the alpha carbon, and they just add onto each other. So understanding these structures are important. The non-polar aliphatic amino acids are glycine, alanine, and proline. The branched chain amino acids are valine, leucine, and isoleucine. Now, when you see aromatic, that is referring to your amino acids that have a ring, okay? And that would be phenylalanine, tyrosine, and tryptophan. And you can see these are your more bulkier amino acids. The polar and uncharged amino acids are similar or look similar to that of the non-polar or the branch chain, but there's a difference because of the composition of the molecules within its R group. So your polar uncharged are going to be your asparagine, glutamine, serine, and threonine. There's only 2 amino acids that contain sulfur. You need to know these: methionine and cysteine. And then you have your charged amino acids, which either can be negative, acidic, or positive, basic, and they are here as well. And you'll see these are even, the positive or basic are even larger in size. Alrighty. Now again, why do you need to know that in terms of its structure? Improper folding can lead to aggregation disorders such as Alzheimer's or prion diseases. So understanding the structure of these amino acids, their 3-dimensional shapes, and how they can form and interact with each other is important for your understanding of their interaction and the role they play with proteins. Okay. Again, amino acids can be classified based off their polarity, charge, and structural features. So using that classification helps us to predict how they'll behave in water, how they interact within the membranes, or even at different pH levels. You'll often see the reference of the pKa values and hydrophobicity index, and that helps understand the acidity and water affinity. That comes into play or has a relevance when we're talking about drug development. So when drugs that you take, your parents may take, are developed based off of understanding the chemical structure and nature of amino acids if it's a protein-based drug. And that helps us understand how it interacts, even if it's intravenous drugs or drugs that have to cross the blood-brain barrier. So understanding their protein composition is extremely important. All right. So our aromatic, your nonpolar amino acids, like valine, leucine, and isoleucine, prefer to hide inside protein cores. So you won't often see them on the outside. They tend to aggregate towards the inner part of a protein, and they are trying to hide away from water. Okay. However, aromatic proteins, amino acids like phenylalanine, tyrosine, and tryptophan, those are your ring structures. They are great at absorbing ultraviolet light or UV light, and they also participate in cell signaling. This is going to come up when we go into the later on in the semester and we start talking about cell signaling. These are a lot of the amino acids that are actually involved in gene regulation and also protein regulation, where phosphates can be added, like tyrosine, tryptophan. So these are important. A lot of times labs use UV absorption at 280 nanometers to estimate protein concentration, and that is a direct application of these aromatic rings. All right. So here you see again tyrosine. It's going to be more polar and hydrophilic. Tryptophan is a little bit more complex because it has that indole ring with nitrogen, and it can engage in hydrogen bonds. So it's going to be more polar than phenylalanine. So here's a question. I want you to pause the video at this point, and I want you to try to answer this question before you proceed. The proteolytic digestive enzyme chymotrypsin cleaves the peptide bonds formed by the carboxyl groups of large, bulky, uncharged amino acids. Which amino acids fall into this category? If you are reading this question and it's a timed exam, the first thing you want to look at is identify what they are asking or the characteristics that they are asking or describing. They say carboxyl group, large, bulky, and uncharged. The first thing that your mind should be thinking is which amino acids are bulky and uncharged. Okay? Example, aromatic amino acids. Remember, we talked about your aromatic amino acids have those ring structures, so they're going to be bulky. That would be your phenylalanine, your tyrosine, and tryptophan. These side chains of the amino acids are large and they're uncharged. So that would be an example of how chymotrypsin's activity would aggregate more towards those type of amino acids. All right, so your polar amino acids like serine, threonine, asparagine, glutamine form hydrogen bonds and are usually on the protein surface, so they're on the outside. Sulfur-containing amino acids, cysteine, methionine, they add extra chemical functionality. So 2-cysteine amino acids can form a disulfide bridge, which help to stabilize a protein structure. And if you remember just a few slides previously, I talked about the tertiary structure or the three-dimensional structure of a protein. You often see disulfide bonds or bridges formed to help stabilize a three-dimensional structure of a protein. So these are very helpful. Disulfide bonds are essential for the proper folding of insulin and antibodies, and if they break, the protein can often lose its activity. So if it's a protein that has an enzymatic function or a protein that is generating or responsible for stimulating hormone activity or a pathway, and it has a disulfide bond there, if there was a breaking of that disulfide bond, that protein would no longer be active or functional. Usually for like cysteine, it has a sulfohydryl group, so it has a pKa of 8.4, and it's predominantly undissociated and uncharged at a physiological pH of 7.8. So if you were to put a molecule of cysteine in solution, it's able to form covalent disulfide bonds with another cysteine. And again, those are helpful with stabilizing the structure, the three-dimensional structure of a protein. Here's another question. At this time in the video, I would suggest you pause it, read the question to yourself, and before you press play to hear the answer to the question, think about what keywords stand out to you to help focus your thoughts in how you're going to attack this question. So it's asking you about the 6th amino acid and beta globin chain of hemoglobin from glutamate to valine. What difference would you expect when you compare glutamate versus valine in these 2 amino acids? Now, you can go back to visually see the structure of glutamate versus valine, but one thing that is important to note between the two is glutamate carries a negative charge on its side chain at physiological pH, so it can engage in high ionic and hydrogen bonds with water. Valine is hydrophobic, so it tends to exclude water. And so this is why when you have that mutation in patients that have sickle cell anemia, their beta chain of hemoglobin starts to kind of fold in on itself and causes the sickling of red blood cells, because that valine is hydrophobic, and so it is not water loving, and it alters the shape of the protein. So amino acids like aspartate, glutamate, they carry negative charges at physiological pH, while basic amino acids such as lysine and arginine carry positive charges. The balance of these charges give each protein a specific isoelectric point, or PI, where the molecule has no net charge. If you all don't remember about isoelectric point, I would strongly suggest you look that up, because that's a prerequisite concept that you should be aware of. So this is a visual picture of a salt bridge that can form in amino acids. So you'll see a change in pH depending on if a molecule or a protein is changed in its environment. There's a change in its environment. So histidine, its structure changes as the pH of a solution changes. And so depending on where this protein is housed or involved in what solution, it will alter its structure. You could have instances where a charge will go from 0 to negative. So how can we measure this or view this? You have techniques like gel electrophoresis that allow you to exploit these charge differences to separate and identify proteins. And this is actually vital in diagnosing hemoglobin variants in liver disease. Okay, so this was the isoelectric point that I was talking about. It's the pH at which the net charge of a molecule in solution is 0. Okay, that is the isoelectric point. So in this diagram here, you can see as pH increases, as you add more hydroxyl, which is the base. Okay, and so this is important in understanding how amino acids can be altered and how they can interact with other molecules. Okay, so why does this matter? Why is this important? How is this relevant to medicine and a practicing physician? Understanding amino acid charge and behavior isn't just academic. It underpins modern medicine. Protein drugs like insulin, monoclonal antibodies, enzyme replacement therapy, all rely on these biochemical properties for stability and activity or reactivity. So understanding this is important. This is not just academic, and I'm just not wanting you to learn this to memorize, but this is going to be crucial into your beginning stages of understanding medicine. Okay, so again, understanding the composition of these amino acids are going to help you, and you'll also see this used in the research labs when you are investigating various proteins and you want to look at their size and their charge. You can use electrophoresis to separate out proteins based off of their charge. All right, so let's talk about variations. We'll switch a little bit. Variations in primary structure of a protein. So the primary structure of a protein is its linear sequence of amino acids. So it's not looking at how it folds or interacts with any other molecules. It is just the primary structure or the linear structure and sequence of amino acids. The linear sequence of the amino acids, also known as the primary structure, can vary slightly among individuals and species. Some substitutions are harmless, while others can dramatically change a protein's conformation or function. So you can see here just the variation. Here's just that sulfide bridge we talked about, okay? And this would be an example of a linear sequence of amino acids. However, removing this disulfide bond here. All right, so a single base, so let me go back for a second. A conservative substitution and like changing an amino acid can go unnoticed, but a non-conservative change in a critical region can result in a disease, okay? And this helps us understand how some regions of a protein are more conserved than others, because in a protein, for an example, in a conserved region, that means, and when I say conserved region, look at hemoglobin. If you look back at globin genes, you have myoglobin, hemoglobin, they're different globin genes, and they are evolutionary conserved in certain regions, but they have a basic amino acid structure that is similar. If you were to induce a mutation, or a mutation occurred in a conserved region, it can be, or a critical region, that would cause or change the function of the protein and can potentially induce a disease or be the cause for a disease. So regions that are critical for three-dimensional structure and contribute to binding sites are called invariant regions. Non-critical regions in a protein are called variant regions. And so it's important to understand this, because if you're doing drug development and you're trying to develop a peptide drug to target a specific protein, for example, that is high in insulin, patients with diabetes, you don't want to cause a change in the invariant region, because that will cause the protein to be inactive or non-functional. This brings us to polymorphism in protein structure. So a single base change in DNA can alter one amino acid, and that is considered a point mutation. This should be something that you all remember from basic genetics, where you have a normal sequence of, let's say, TTC. On the mRNA level, it is AAG, and it produces the amino acid glycine. If you were to produce a mutation, or a mutation occurred and it's silent, that means you changed like a C to a T. It produces the amino acids, mRNA structure AAA, but it can still produce the amino acid lysine. That's considered a silent mutation. However, there are two missense types of mutation that we want to talk about: conservative and non-conservative. This is where you change that T, that second T, to a C. Well, changing that second T to a C produces AGG, which produces the amino acid arginine instead of lysine. That is a conservative example. A non-conservative example would be changing that T to a G, which would then produce threonine. Different amino acids. Okay, very different amino acids. So, for example, in sickle cell anemia, the substitution, as I stated earlier, of valine for glutamic acid in hemoglobin causes red blood cells to deform into a sickle shape. And that then alters how it interacts with oxygen, and it actually causes pain, because patients that have sickle cell anemia and have the sickling of their red blood cells makes them kind of sticky, and so they aggregate into the vasculature. It causes them to have severe pain throughout their body, and also oxygen deprivation. So doing that causes what a lot of patients that have sickle cell anemia call a pain crisis. Okay, now it gives them immunity to malaria, which is an example of evolutionary balance, but they still experience side effects from having that one mutation. And so understanding polymorphism is just showing you that one mutation can have a widespread effect that just does not have one symptom but multiple symptoms, and it can be serious. Okay, so this is just visually showing you the normal red blood cell versus the sickled red blood cell, and that small change, that one base change from glutamic acid to valine, has a very serious side effect. Okay, so let's transition a little bit and pivot into protein families and superfamilies. Proteins that share similar structures and functions belong into what we call, are grouped into families. And usually it's often based off their origin, and they share some type of homology. So their sequences are very similar, and they only have little portions of their sequence that differ and make them unique. Through gene duplication, mutations, these families of proteins diversify into what we call paralogs, and those are proteins that are related but have different functions. Myoglobin and hemoglobin are classic examples of this relationship. They're both foundational globin genes, but they operate differently. This explains why fetal hemoglobin binds oxygen more tightly than adult hemoglobin, which is an adaptive, a life-saving adaptation during development, right? So a fetus is living in an aqueous environment, and so it has to be able to have a tighter affinity for oxygen. But once that fetus is born and starts to grow and mature, it transitions to adult hemoglobin, and it does not need that tight affinity for oxygen. Okay. The superfamilies, let me actually go back for a second because I want to make sure I point out when you have large amounts of homologous proteins, so not just one or two, but a larger amount, we call those superfamilies. So a family of proteins may just be maybe 3 or 4. Superfamilies can be 10 plus proteins that are very similar but have small differences that make them unique and distinct. So here you have myoglobin, okay? And what I like about this picture here, if you all can see, is you have myoglobin here, and then you have the different chains of hemoglobin, beta, alpha, gamma. But notice there are certain regions that are conserved, similar amino acids, and then there are other regions that are different, okay? But through these gene duplications and other evolutionary processes, you have certain regions that don't change, and there are what we call conservative substitutions, because it's important that the basic structure of these globin genes, which are oxygen binding molecules, do not change. All right, so tissue and developmental variations in proteins. So within an individual, there are different isozymes of a protein that can be produced at different stages, okay? Different tissues may express different unique isoforms of enzymes. That's what isozymes are, okay? And a great example is creatine kinase. And it exists as CKBB in the brain, so creatine kinase in the brain. And then in the skeletal muscle, it is CKMM. And in the heart, it's CKMB. So you have different isoforms or variants of creatine kinase. Now, the importance of understanding the variations of these different enzymes is, for example, if someone has a heart attack, doctors can go in and look for the isoform associated with the heart. So if a patient has a heart attack, right, but they maybe don't realize it or they're not sure, the physician is going to draw the blood and send it off to the lab. Well, what indication or marker is going to let them know that there has been a potential heart attack? They're going to look for elevated levels of creatine kinase MB, because the creatine kinase MB is the isoform associated with the heart. And high levels of that are a key marker for myocardial infarction that helps physicians confirm that a heart attack occurred. So again, I hope that this also helps you understand the importance of what we're talking about. It's not just for academic purposes, but understanding that proteins exist in isoforms, and the different isoforms relate back to the different tissues that they're present in and how they function, are also used clinically in medicine, in the hospitals, to determine disease progression and whether it even occurred at all. Okay, so here you have the fetal hemoglobin, here's the adult hemoglobin, and remember, the fetal hemoglobin has a higher affinity for oxygen than the adult form. And this is why fetuses are able to thrive in an aqueous environment as opposed to adults. Okay, so here is the what I was mentioning, the different forms of creatine kinase: B form, M form, and MB. There are other forms like mitochondria, and then there's a universal form. But ultimately, these different forms are important for their function and structure in different tissues. So even though an enzyme can operate in different tissue, its basic function should be the same. It just operates differently. So the creatine kinase that we see in the brain has the same function in the heart, but it operates differently in the tissue. Cyclic AMP is another example, right? Some protein isoforms respond to the same hormone but differently. Cyclic AMP has 9 different isoforms. So here is a diagram showing you how you can identify the different creatine kinase isoforms, whether it is the heart or the brain. There's the BB, brain, and then you have the MM for the skeletal muscle. All right, so I want you to pause at this point and read this. I kind of talked about it already, but this is a clinical vignette to help you understand the patient angina. And it also helps tie in creatine kinase in the myocardial infarction, which is the heart attack. All right, we're almost done. So species variations in insulin. So insulin structure is highly conserved across species, but even small amino acid differences once caused allergic reactions in patients who receive bovine and porcine insulin. So this is a reason why there was this influx to figure out a way to create human insulin or synthetic insulin using the human gene for insulin, but in bacteria, because there was a lot of allergic reactions or immune responses that were induced when patients were using bovine insulin. So being able to create recombinant human insulin eliminated that issue. And that was in part due to our understanding of the chemical nature and composition of the protein insulin. But this is also a great example of how understanding amino acid differences guides safer pharmaceutical design. So I want to point that out. Understanding the amino acid differences in the chemical composition of various proteins helps us to develop drugs that are more efficient, that hopefully are safer for the patient and also more effective. So here is a schematic of the structure of human insulin. And it shows the residues and how these residues interact with each other to form this 3-dimensional shape. For example, there are multiple cysteines, amino acids in insulin. And we already know if there are multiple cysteine amino acids, they can form disulfide bonds, as you see here. At least 3 disulfide bonds in the amino acid, in this protein structure of insulin. And this allows it to have its 3-dimensional shape. The other residues allow proteins to bind to the insulin receptor. So understanding its amino acid structure also allows us to understand how it can interact with other proteins and also receptors. So take the time to pause this again and to read this clinical portion, just helping you understand the history of insulin and how it has evolved over time. Again, initially it was bovine or beef insulin that was used to treat patients that had diabetes. But because of the influx of immune responses and allergic reactions, they were able to examine the structure of insulin, identify the residues that are variable and that are able to act as antigens and stimulate the formation of antibodies. And then they were able to use recombinant techniques to create recombinant human insulin. And so this, again, is an important an advancement in the industry, because this also allowed them to be able to mass produce insulin at a higher rate than they were before. All right. So after proteins are synthesized, they often undergo modifications like glycosylation, phosphorylation, or lipid addition. These chemical changes are important because they help fine-tune protein function, stability, and localization. We call these post-translational modifications. So after a protein has been synthesized through translation, it can further be modified by adding different functional groups to the amino acids within the protein, and it allows the protein to take on different conformations. But some of these modifications can also be regulatory. So it can anchor a protein, it can enhance an interaction with other molecules, and it also can target a protein for degradation. Okay, so what is glycosylation? Glycosylation is where you add a small modification of a carbohydrate chain to a protein, whether it's N or O linkages, which we learned about in chapter 5. And so an N-linked oligosaccharide attaches to a cell surface protein, and it can protect the cell from proteolysis or an immune attack. If you have an O-linked oligosaccharide, that's let's say attaching oligosaccharide to serine or threonine can help with secreted proteins. Acetylation or prenylation is often used where a protein contains a covalently attached lipid group that interacts hydrophobically with lipids in the membrane, and many of these are also involved in regulation. And then you have your regulatory modifications. This would be your phosphorylation, your acetylation, your ubiquitination, methylation of specific amino acid residues. And this can alter the activity of a protein, okay? You have reversible acetylation on lysine residues or histone proteins that can cause DNA to condense or decondense. Methylation of an amino acid residue can affect gene expression, so it can turn genes on or off. And so this indirectly affects protein, but you also have phosphorylation, acetylation. This is where a phosphate group is added or a acetyl group is added, and that can either turn on a protein or it can turn off a protein. It can cause a protein to be arrested in the cytoplasm, or it can cause a protein to be activated and trigger a cascade, thereby going into the nucleus and regulating gene expression. Ubiquitination can stimulate a protein to undergo degradation by adding an ubiquitin to the protein. So all of these, again, are important, and they are also utilized in drug development. Okay, so there are certain drugs that induce protein modifications. For example, paclitaxel, nocladazole, those are two very popular drugs that are used in cancer, and they work by adding a phosphate group to BCL2, and it stimulates cancer and is anti-apoptotic. So oftentimes in cancer, in the field of cancer, you have a lot of drugs that are protein kinases, and they have become a very important drug target in the pharmaceutical industry. Currently, there are about 25 drugs that target protein kinases, and they've been approved clinically for use in cancer treatment. One of the big pharmaceutical companies, Pfizer, has a JAK3 inhibitor called tafasitamab, and it is approved by the FDA. It was approved back, I think, in November of 2012 for treatment of rheumatoid arthritis and other inflammatory diseases. So I wanted to point those out because I want you to be aware that while we're learning about different protein, how proteins can be modified, and you may feel like it's boring, it's important that you know as a future physician, because you're prescribing these drugs to patients, and you need to know how they're interacting once it goes in the body. One of my favorite shows I'll tell you guys about is House. House is a great show because it helps you understand how it is important for physicians not to just memorize, but to really understand in depth what they're doing, because there are going to be instances where patients have a disease or symptoms that are hard to understand. It's not as straightforward. You can't go to the Merck index to understand or Google it. You have to know your information, and part of this comes in understanding how the chemical composition of amino acids and proteins work and how they interact with each other. Extremely important. There are some drugs that deliberately target modification pathways, and so if you don't understand this, it can be a very, very difficult path to just understanding basic concepts in medicine. Alrighty, so I want to point this out. This was written by a medical writer, Lindsay Tanner, but she says, why is elephant cancer rare? This was actually given to me by one of our former chairs of our department, and he came to my office and gave it to me. He was so excited to share this story with me, But as I began to read it, I thought how amazing this is that we don't actually look into this. And scientists started looking into elephants because they don't get cancer. It's actually very, very uncommon for elephants to get cancer. And the question that they asked was why? Why do elephants have a very uncommon prevalence for cancer? And it is because, in part, they have multiple copies of a gene called, a protein called P53, which plays a major role in cancer. So I want you guys, if you want to read more up on this, please definitely Google this article. It's a great read. And I've been telling students about this for years. The former chair gave it to me about, he's been gone for at least 7 years now. But this was a great article. And I think you'll find it interesting. All right, so as I'm preparing to close out the lecture for Chapter 6, I want you to, again, I provided the clinical comments from the textbook in case you don't have it, but I want you to read upon the clinical comments for Will sickle and diabetes and angina. And the reason is this is going to help you understand how we started off with them in the beginning of the chapter, and then tying back to what we've talked about throughout the chapter with the patients and some of the symptoms that they experienced. You're also going to see this come back up for those that have to take the comp exam. We're going to use some of these concepts in your comp exam as ways to tie these concepts back to what you're doing. So make sure you take the time to read through these clinical comments and tie back in the biochemical aspect to the clinical aspect, because that's a really important concept that you all need to be able to connect.